Good evening, everyone. I'm Kelly Ritalik, the Director of Genetic and Genomic Programs for Angus Genetics Inc, and we're excited to have you here tonight at the 7th installment of the Angus University webinar series. Tonight, our group at AGI is excited to present to you some research that we've been working on for quite some time. But before we get to that, let me ask you a question. Have you guys ever bred a group of females? Guaranteed yourself that you saw that one female get bred. She passed her 3 weeks, but as soon as you're tuning her out to pasture, you can see the bull chasing her. Well, what happened to that pregnancy? You're almost 100% that you got her bred. So what happened from the time that you turned her out, from the time that you bred her to the time that you turned her out? Well, our geneticists here at AGI may have an answer for you. And that answer could be haplotypes that affect fertility. But more on that later. Dr. Duc Lu is going to join us later on in the presentation. He is a research geneticist here at AGI who's been working on this fertility haplotype research for some time. We're excited to present this information, so let's get started. Getting cows pregnant is one of the main drivers of economic sustainability in the beef cattle industry. In 2008, the Beef Site reported that $1.1 billion was lost in revenue due to infertility. So from our standpoint here at the association, we've tried to come up with tools such as scrotal EPD, heifer pregnancy EPD, and even $M, the maternal index, to be able to tackle these things associated with fertility. But because we know that it's of utmost importance to you as breeders, we wanted to go a step further. So in order to do this, we took a step back to look at the information we do have and how can we better leverage that for you. So Angus has a large database of genotypes. Currently in your genetic evaluation, we have 868,000 genotypes that come from multiple different sources. So currently right now you have available the Angus GS test and the HD50K for Angus test that's available through our contracting partner suppliers. With that, we have many older genotypes in our database as well, such as our GGP-LD, GGP-HD, and I50K genotypes that have been collected since 2010. So with these genotypes, we currently do several things. So the first thing that we can do with our genomic testing technology is that we can basically track physical characteristics such as coat color or horn-polled genes. We can also track specific mutations like our genetic conditions that we have in Angus cattle, as well as use those genomic tests for parentage verification, and finally use them for genomic selection through the inclusion of that genotype into the EPD. But when you send in that $37 for that genomic test, what are we actually getting back at the association that helps you to basically select cattle easier and more accurately with genomically enhanced EPDs? What we get are a file of SNPs. It's actually abbreviated SNP for single nucleotide polymorphism. This is basically an identified location in the genome on specific chromosomes of animals of the same species where specific alleles are variable. So if we break down single nucleotide polymorphism, we can take it one word at a time. Single refers to a single location. Nucleotide confirms to the 4 base nucleotide alleles, A, T, C, and G, and polymorphism means morphed, or basically that those A, T, C's, and G's are different at one location. So when we get the results back from the lab, this is what we see. As you can see, it's a lot of work to be able to take what we receive back from that genotype and filter it in through our genetic evaluation to get back genomically enhanced EPDs. But as you can see here, we also get a lot of other information, and how can we leverage that for you? Additional use of Angus genotypes could be used in several different capacities. So we know right now that we can track physical characteristics, that we can use them for parent verification, and also we can use them to enhance EPDs for genomic selection, but what other opportunities exist? At this time, I would like to invite my colleague, Dr. Duc Lu, a research geneticist here at AGI, to talk about another opportunity. Dr. Lu?
Thank you, Kelly, for the introduction. So Kelly already explained to you the basics about SNPs. I'm going to talk to you a little bit about haplotypes. But first, let me present these 3 DNA segments to you. So each of these DNA segments represents an animal. Let's focus on animal 1. So we all know that for a given DNA segment, there are 2 DNA strands. This is strand 1 and this is strand 2. And for a given SNP position, for example, this animal 1 here at SNP number 1, there are 2 alleles, one on each DNA strand. So this animal has 2 different alleles at SNP number 1, and he is heterozygous at this SNP position. At the next SNP, he has 2 identical alleles at SNP number 2, B and B. So he is homozygous at his SNP location. Okay, now let's focus on this region from SNP 14 to SNP 23. This is a haplotype. A haplotype is a string of consecutive alleles on the same DNA strand and tend to be inherited together. The length of a haplotype could be various from a minimum number of 2 alleles to the maximum of the length of the chromosome. And for this example, we have a 10 allele haplotype right here. And for a region on the chromosome, there are 2 haplotypes for each animal. This is haplotype 1 on one strand of the DNA, and this is haplotype 2 on the other strand. And if you look closely, you will notice that these 2 haplotypes are identical. So this animal is homozygous for this haplotype at this region, just similar to he is being homozygous at SNP number 2. Now, animal 2, for the same region, from SNP 14 to SNP 23, there are 2 haplotypes as well for this animal. And these 2 haplotypes are different. So animal 2 is heterozygous for the haplotype in this region, similar to the concept of heterozygous at SNP number 1 for animal 1. Similarly, for animal 3, he has 2 different haplotypes, and so he is heterozygous for this haplotype at this region. Now, if I'm interested in the haplotype in the green box, let's call it the green haplotype, and I would like to know how often this green haplotype occurs in my population. So I'm going to count the number of this haplotype in my population. So animal 1 carries 2 copies of the haplotype. Animal 2 carries 1 copy of the green haplotype. So there are 3 copies of this green haplotype in my population. So the total number of haplotypes for this region in my population is 6, because each animal has 2 haplotypes, right? So this table summarizes the count of the green haplotype in my population. So animal 1, 2, and 3 with 2 copies of the green haplotype, 1 copy of the green haplotype for animal 2, and 0 copy of the green haplotype for animal 3. So the total number of green haplotype in my population is 3, which is the sum of 2, 1, and 0. And the total number of haplotypes for that region in my population is 6. So my population has 3 green haplotypes out of 6. So the frequency of this haplotype in my population is 50%. So this is how you calculate frequency of a given haplotype, any haplotype across the genome. Well, let's say If I call the green haplotype a wild type and I give it a small w, then any other haplotype other than the green ones, I put them in a group I call big W. Then animal 1 carries 2 copies of the green haplotype, so he has 2 little w's. Animal 2 carries 1 copy of the green haplotype and 1 red haplotype, so he is a big W, little w. And animal 3 carries 2 red haplotypes, so he is 2 big W. Now, let's focus on this animal 1 and 2. Animal 1 is a homozygous for this haplotype, right? So he has 2 copies, 2 little w. And animal 2 is a carrier because he carries 1 copy of the green haplotype. Let's look at this example. If I have a cow that is a carrier, he has a green haplotype and a red haplotype. And a sow, he is also heterozygous, so he carries 1 green haplotype and 1 red haplotype. According to Mendelian law of segregation, all the eggs coming from this dam would have 50% chance of carrying the red haplotype and 50% chance of carrying the green haplotype. Similarly, all the sperm cells from this sow has 50% chance to carry the red haplotype and 50% chance of carrying the green haplotype. Let's call red eggs, green eggs, red sperm cell, and green sperm cell. Okay? So if these 2 individuals made it to each other, then if a red egg conceived with a red sperm cell, then the progeny is a homozygous red. So he carries 2 copies of the red haplotype. In other words, he's a non-carrier of the green haplotype. If a red egg conceived with a green sperm cell, then we have this animal who is a carrier of the green haplotype. And if a green egg conceived with a red sperm cell, we have another heterozygous for the haplotype. And if a green egg conceives with a green sperm cell, then we have this homozygous green. So he carries 2 copies of the green haplotypes. So 4 kinds of combination, but actually only 3 phenotypes, 3 kinds of genotypes. Homozygous red, heterozygous, these 2 together, and homozygous green. So you will notice that the probability for the progeny to be homozygous red is 25%. Probability for the progeny being heterozygous is 50%. And probability for the progeny to be homozygous green is 25%. Assuming that you have 1,000 matings like this between the 2 carriers, then you would expect to see 250 homozygous red, 250 homozygous green, and 500 heterozygous, both green and red. So that is perfect math, right? If it works perfectly like that, you would see that perfect number. But in reality, the numbers could be deviating from the expected number. So instead of having 250 homozygous red, you might observe, let's say, 245 or 255, for example, as long as the deviation is not significant. So in that case, if you have the similar number of homozygous green, let's say 250 or a little bit less or over, then you would expect that this green haplotype behaves normally. then you would have that 25% of homozygous red, 50% of heterozygous, and 25% of homozygous green. So that is a normal situation. But situation like this could happen too, where homozygous green just don't exist in the population, or for some reason they disappear, or they exist at really low frequency. Let's say we expect to see 250 of such homozygotes, but we observe only 1 or 2. So there is that missing homozygotes in the population. Let me ask Kelly. So what do you think about what would be the cause for that missing homozygotes?
Yeah, so this is exactly what you're looking for in this population, right?
Yes.
This pattern in which we don't see those green homozygotes, and a reason for that could probably be embryonic death, right?
Right.
2 of these animals, maybe the 2 green egg and green sperm are basically meeting up together. That embryo, right, that's basically being grown inside that dam doesn't become viable. That embryo dies off, so that calf's never born.
Right.
The calf's never born, right? We can't genotype that animal.
Right.
So you've been looking for this pattern in our data for quite some time, right? You've been using our genotypes, looking through all our genotypes that we have in the database. How many of these types of regions do you currently see in our population?
We actually found a handful of haplotypes that behave like the green ones, but they are at different frequency. Some are low, extremely low. Some are a little bit higher. From a research perspective, I would say that I would like to investigate all of them to see what happened. But from production-wise, what do you think? What would you think would be like a frequency that would be alarming to breeders?
Yeah, so when you talk about frequency being at variable levels, you're talking about frequencies, what, in the 1 to 5% in the population?
Yeah.
Yeah, so if we think about frequencies of haplotypes in the 1 to 5% of the population, let's just take a haplotype frequency of 1, right? If we have haplotype frequency of 1, that would mean we have a carrier frequency of 2, correct?
Exactly.
So let's say we have 900,000 genotyped animals across the population. Okay, we have 900,000 animals, we have this haplotype carrier frequency of 2%. That would mean we would roughly have 18,000 carriers in the population?
Yes.
So I would definitely say 18,000 carriers is a significant thing that our breeders would have to breed around.
Yes.
Would you agree?
I do. And actually, when we say low frequency, it could be low in the entire population, but it could be high in some certain herds.
Sure. So because I use certain sires, right, these frequencies may be higher in my own personal herd because just luck of the draw.
Right. So for that reason, it could be alarming for some breeders, but not for others.
Sure. Yeah, that makes total sense. So another question that we get often, right, are what are the differences between what we're calling a haplotype, right, and what we've experienced in the past with genetic conditions?
That would be great to distinguish.
Yeah. So even though the mode of inheritance is very similar, as we see this kind of recessive mode of inheritance being passed down, there's quite a few glaring differences between haplotype and a genetic condition. So when we think of genetic conditions, we're talking about an individual mutation that we have identified, right? So we can go into the population, we can look for a specific insertion, deletion, maybe a change in that DNA variation, and we can indefinitely say that this animal has a genetic condition or does not. The other thing with our genetic conditions that we experienced back in the early, near 2008 to 2010, is that they're in much higher frequencies than Dr. Lu was talking about before. We saw frequencies of at least of greater than 15% when those genetic conditions first came on the scene. And then with that, with our genetic conditions, many of them are lethal, right? But they have a greater financial impact in our herds because those cows are carrying those calves to full term. They're actually giving birth to those calves that then are usually not born alive. And then you end up with not only a dead calf, but an open cow. When we talk about haplotypes, what Dr. Duc Lu is doing is he's basically identifying a phenotypic pattern, right? He's basically identifying a haplotype pattern that is associated with some type of phenotype. So when we talk about this, we're not identifying, right, the causative mutation when we identify a haplotype. So we're not identifying that insertion, deletion, etc. Rather, we're identifying a pattern of these haplotypes that are then, what we are doing internally, are trying to associate it with a particular trait. Whether that haplotype is being associated with something like fertility or growth, basically we find the pattern and then we try to find what is that haplotype causing in our population. With that, as Dr. Lu said, these are happening at much lower frequencies in the population. We see ranges in frequencies of about 1 to 5% for these haplotype regions that we're trying to focus in on. Similar industries, other industries have done similar research in their own kind. So if we take the dairy industry, for instance, they've been using fertility haplotypes since about 2011. So when you look across the haplotypes, for instance, that we see in the Holstein breed, they would have very similar frequency estimates, carrier frequency estimates of between 1 and 5%. And then with that, when we compare the financial loss between haplotypes and genetic conditions that we saw from 2008 to 2010, there's a lot less financial loss associated with these haplotypes, because if they are indeed associated with early embryonic lethality, basically we can come back and we can rebreed that female, and if we don't mate those two carrier animals together, we're not going to have an incidence of basically stacking up these two haplotypes. Even if we came back and made that same carrier-to-carrier mating, there's only a 25% chance that this embryo is going to receive two red haplotypes from the sire, two green haplotypes from the sire and the dam, and it has a 75% chance of being completely normal otherwise. So what's the opportunity for Angus breeders around these haplotypes? So the first thing that we want to do is we want to leverage this unique information for Angus breeders who a lot of other beef breeds and a lot of other people in the beef industry really aren't in a position to leverage. Because you all as breeders have been so great at buying into technology, at genomic testing your animals, and really participating in basically genotyping, we're able to leverage your huge genotype database to be able to go in and look at some of these things before they become so high frequency in our population that they're going to cause greater associated fertility losses. So really the end goal out of this entire research project is to create a breeding management tool for you. So the first thing that we need to do is the process that we're undergoing right now. We need to identify these regions of interest, and we need to validate these regions of interest with real phenotypic data. And the reason, and the types of data that we're looking at currently are around fertility. If we are truly finding these, if we're truly missing these homozygotes because of the fact that we're losing those embryos early on in pregnancy, we need to try to take what data you all have sent into us from breeding data, from calving records, etc., and try to use that to find a phenotypic pattern associated with this haplotype region of interest. The second thing we want to do after we identify and validate is we want to make this information accessible to you. We want to make this information accessible to you so you all can mate around these nuances in the population. And ultimately, if we can mate around these nuances, basically we can decrease the overall frequency of the haplotype in the population, and moving forward, continue to monitor frequency of unknown haplotypes that we may not know today. A lot of times we get asked the question as to why we're identifying or why we're thinking about identifying animals if the frequency of these haplotypes in the population are so low. Well, the answer to that question is, as we spoke about earlier, if we leave these unchecked and we leave them out in the population unchecked and just allow you to continue to breed animals in a spurious manner, we could basically increase the frequency of those haplotypes to a level that then would be very detrimental to the population. The other thing we want you to understand is that we truly believe these are breeding management tools. And because they are breeding management tools, there's no registration restrictions around these tools. The thought behind this is we would never restrict your animal from getting registered just because it may have a poor scrotal circumference EPD, for instance. Scrotal circumference EPD is also a breeding management tool, and just because you may be poorer than breed average in that category wouldn't cause a demerit on your animal for that animal not to be registered. Because these are breeding tools and we feel that they could be very beneficial to your breeding program, we want to treat these in the same manner. So let's recap what we know, okay? Dr. Duc Lu explained to us that we have one region of interest in the genome right now. He's identified a handful of regions that he wants to take a look at, but the one we're zoning in on right now is a research haplotype of interest. And the reason it's a research haplotype of interest is because it goes against the normal population genetic theory, Mendelian sampling theory, that basically we do not see 25% of our progeny when we mate two animals together that have two carrier dam and sire together. We do not see this 25% portion of progeny getting genotyped with that WW allele. So the real question is, where are these missing homozygotes? So where are these 25% of our animals that aren't showing up with this particular haplotype status? So the first reason we've talked about numerous times throughout this presentation is that these haplotypes are affecting, right, our embryo development, and because those embryos aren't viable, the progeny are never born, and if a progeny is never born, we surely can't genotype it. Or, on the flip side, it could be the fact that these animals are being selected by you all as breeders to not be genotyped for some reason. Some reasons for that may be around the fact that those calves are born too large, and because you guys know that they're too large, you're not going to keep them for breeding bulls or replacement females. Another reason could be the fact that the calves just fail to perform up to weaning age, and so because they're lacking performance at weaning time, you cull those animals from your herd. They're not kept as yearling bulls to sell in your next bull sale or as replacement females because they're not living up to the rest of the population, and so for that reason, you're not investing the $37 to genotype them. That's the question that we're going over internally, and that's the question that we want to answer before we would ever report this information to you all as breeders. So if we're not ready to report those haplotype statuses yet, what's next? So obviously Dr. Duc's research shows a lot of promise, and we really want to continue to pursue this research actively. So currently AGI is diligently trying to validate this haplotype and analyze the discrepancies within the population. Currently we've been trying to leverage all of your fertility data that's in-house. That's going to be heifer pregnancy records. It's going to be cow fertility records that you all have sent in. We're looking at cow calving records, and we're trying to figure out when cows leave the herd and why did they leave the herd. And we're also looking at correlations between this haplotype with your heifer pregnancy EPD. But to be honest, up until this point, we haven't found our smoking gun. We haven't been able to confirm that indefinitely, yes, this haplotype is affecting early embryonic lethality. So where does that leave us? That leaves us with you guys, right? We need your help. We need your assistance to be able to move on and move forward with this fertility haplotype. So what do we need from you? Well, we need additional data. When the dairy industry confirmed some of these haplotypes in their database, they were basically using large databases with basically millions upon millions of fertility data points, conception records, breeding records. And in our database, while I'm quite fond of the fact that our members have sent in over 100,000 heifer pregnancy records, it's still just not powerful enough for us to really truly track these patterns in your overall population. So what we're asking of you is that if you have breeding records that include all service records on either your heifers or your cows, if you have breeding records that are basically lined up with a preg check record and other subsequent service records, please let us know. With that, as you're going through and you're reporting your calves for this year, if you have calves that were born but maybe died before weaning, perhaps failed to live up to their genetic potential at weaning time, make sure you let us know if you culled those animals early. We want to know why those calves weren't born, or if they were born, why those calves aren't showing up later on in life. With that, cow disposal codes are a really key way that could help us with this research. Currently, right now, we have cows that will get calves reported to them when maybe they're 2 years old. We'll see that they don't report a calf at 3 years old, and maybe they'll show up at 5 or 6 years old. But in the interim, what's happening to those cows, right? Did they fail to calve because of the fact that they came up open one year? Or did we just not get the birth or weaning weight record reported on that calf? That type of information allows us to better quantify what's actually happening when we see these patterns in your genotypic data. Next month, right here on this webinar, you'll actually hear about the inventory-based AHRR program that's launching next month. This program is a first step and could go a long way to support this type of haplotype research in the future. Another key point that we could look at is our embryo transfer records. Currently, right now, the association isn't collecting a database of embryo transfer records, but it could be very useful to be able to look into your all's embryo transfer records and see which embryos came to fruition and actually resulted in a live calf, and which embryos may have not have resulted in a live calf. If we can go and we can leverage all those types of records that you guys may be sitting on, that would allow us to go in and further validate this research and hopefully get these types of tools in your hands earlier rather than later. So the overall goal of this project would be to identify haplotypes that affect fertility, allow you all an intimate knowledge of the haplotype status of your individual animals, combine that with our heifer pregnancy EPD, as well as you all continuing your great management practices, breeding practices, feeding practices that you do at home so that we together can get more calves on the ground. So with that, I know that we've had quite a few questions come into our chat box as we've been talking for the last half hour. So I want to invite my colleague, Dr. Duc Lu, back up to join me here so that we can answer some of those questions for you. Yeah, so one of the first questions that we have here is around this idea of whether this haplotype is tracking poor performance, maybe poor weaning weight, right? Or is it truly tracking fertility? So does this mean that a particular haplotype would be, that would be good for weaning weight would automatically be bad for fertility?
We did some association for this haplotype with weaning weight and birth weight and post-weaning gain, but it did not indicate any association between the weaning weight and fertility.
Sure.
So far for now, there's no smoking gun there.
Yeah. So Dr. Duc, you did those association analysis. So basically not only did you look at our fertility data, but you also tried to associate this particular haplotype with our growth data, correct?
Yeah.
Right. So one of the things that we know about haplotypes is haplotypes just aren't significant for fertility, right? There's haplotypes all over the genome that are controlling many, many different traits. Exactly. And so understanding whether or not this haplotype is associated with high or low growth, right, would be very imperative for us to know as we move forward.
Yeah. So we've been working on that too.
Yeah. So another question that we have here is, what use could embryo transfer be in determining or assisting in the evidence of these haplotypes? So obviously, as we're mating animals together, right, to create embryos, a lot of times we could run into situations where we're mating these two types of carrier animals together, right? And if we're mating those two carrier types of animals together to create those embryos and then placing those embryos into those females, if those embryos are then lost, right, early on in pregnancy, that could be really valuable information for our research group to tackle.
It is actually a very important piece of information for this research, if we do have that data.
Yeah. It'd probably even be really great if we could go in and, let's say with our breeding records, right, if people actually had ultrasound preg check data, right, and they could actually tell us when and if that pregnancy was actually viable and at what time.
Exactly, yeah.
How do fertility haplotypes and inventory-based reporting work together? So this is a great question, and you guys will be hearing a lot more about inventory-based reporting coming up. But basically, these two things can work together because it basically gives us a whole record of every single herd, right, that would be included in inventory-based reporting. So sometimes we see individuals come in and they may selectively report a group of calves, right? Maybe they only report their top 70% of their calves, and the bottom 30% that they sent off, right, to the feedlot, they don't send that data and those records into us. So when those females come in year over year, those ones that you didn't send in, those females come up and they're basically missing for that year. And we really don't know why they're missing. They could be missing because of the fact that you sent those calves to the feedlot and you just didn't report their weaning weights. Or are they missing because they didn't calve that year? They fell out of your calving season. They didn't get the opportunity to get bred, or maybe you tried to breed them and they came up open later. So with inventory-based reporting, we're going to get a snapshot of everyone's herd each and every year that would allow us to basically go in, identify why if a cow calved, and if she didn't calve, why didn't she calve, right? Was she open? Did she have a fertility issue? Was she culled for a non-fertility reason? Or were you just not reporting the calves because those are the ones you didn't want to keep?
Right. Yeah. So the next question is, given that they have found in Holstein, what percentage of reproductive failure do you feel we will be able to eliminate through the identification of fertility haplotypes? So in order to answer this question, we don't have an exact number so far. But if you look at that square box that Kelly presented toward the end of the presentation, you will see that those homozygotes green do not exist in the population. So what you see right now is the open rate would be 25% less than what it should be if we remove those
Carrier by carrier matings.
Carrier by carrier mating. So you can reduce that by 25%. Like perfect math.
But we all know nothing's perfect in animal breeding, right?
Yeah.
Yeah. So another question that came in is, could producers already be selecting against this haplotype unknowingly by culling cows that consistently come up open, calve late, have to get rebred, or have low performing calves? And the same could be said for the sire side. So this is an interesting question, right? Could individual producers actually be culling these fertility haplotypes out of the herd just because those cows are coming up open, you're having to breed them more than one time, or the cows are just low performing? And I would say there's definitely some room, right, for selecting against this fertility haplotype by getting rid of those problem animals. I think the one thing people have to be aware of too, though, is that there's a lot of environmental reasons, right?
Yes.
Why pregnancy rates would fail.
Yeah.
So one thing that we talked about earlier is that if an individual producer really wanted to take steps today, right, to avoid having these types of haplotypes in their herd, would be to pay attention to their inbreeding coefficients.
Yes.
Right?
That is an alternate.
Yeah.
Yeah.
Just kind of another pathway forward until we get these haplotype statuses out there for the membership to use. Obviously, avoiding using the same common ancestors on both sides of that pedigree would help us to reduce the risk of having these types of recessive type traits being passed on.
Yeah.
So the next question we have here is, given what they have, do you have haplotypes marked that are good for fertility or bad for fertility?
Well, we call it haplotype affecting fertility. Well, it depends on how you look at it, right? If you think that this is something that you can manage to avoid mating between the two carriers, so you could improve the fertility. So you can look at this as kind of like a good thing. Yeah. Yeah.
Yeah, it's absolutely an opportunity for our members, right?
Yeah.
Because these things that we weren't able to track before, they actually could begin to track. And if they can avoid some of those matings, right, they're going to avoid losing 25% of their embryos due to the fact that these haplotypes can be mating up.
Yeah.
So while in most cases when we're tracking fertility, we're looking for the missing, right? And if we're looking for the missing, those haplotypes are probably having a negative effect, but there's probably haplotypes out there too that are having a positive effect. They're just a little bit harder to track.
Good point, because let's say this haplotype, assume that validated,
Yep.
it is impacting fertility, but we still need to do research to prove that it doesn't have impact on other traits.
Exactly.
Sometimes a haplotype is bad for something, but it might be good for other things, for other traits. So there should be a balance in managing this haplotype in the population.
Right. And that's why we encourage people to manage it, right?
Yeah.
We wouldn't want someone to, just because an animal may be a carrier for one of these haplotypes, actually using them as a breeding management tool rather than just purging those animals from the herd,
Exactly.
right? Allows you
Yeah.
to keep those animals in your herd that have really good genetics, right?
Yeah.
You selected them, you kept them in your herd for a reason. So by doing that, we can actually have the best of both worlds.
Exactly. And Angus has been doing really well so far.
Yeah. Will haplotypes be identified in both dams and sires?
Until now, this haplotype is not sex specific. So it is in both SARS and DAM.
Sure.
Yeah.
So basically anyone who, right now what we're kind of going over is, can we basically make a haplotype status call on anybody, right, who's been genotyped over the past several years?
Yeah.
Right? And we're still continuing to validate that and do those types of things. But yeah, there's nothing that's sex linked here. So we should, in theory, right, be able to call it on
Yes.
both the sires and the dams.
Yeah.
Our next question here, is it viable to perform experiments with animals that have the lowest green WW populations, i.e. create embryos and develop in recip cows?
This is a perfect world. So in a perfect world, we would like to carry out such experiment. It is just like one step to validate the haplotypes.
Yeah.
So how to put out a call for that?
I think you're kind of alluding to the fact that this would be an expensive thing for the association to take on ourselves. And
Yes.
so that would be something that we would probably need to get help with in order to do those types of things, because obviously creating those types of embryos to basically then test whether or not that embryo is going to be viable, that would probably take some genotyping of those embryos and things like that. And we know with genotyped embryos, being able to get high call rates on the genotypes from those embryos, the call rates go down a little bit.
Yeah.
Right.
Yeah.
So being able to accurately call haplotype status on an embryo might be a little bit challenging.
That would be challenging, but if you follow, let's say, if you make the two carriers to each other, and if you just check the status of the embryos,
Sure.
if they survive throughout the pregnancy time, that would be a good thing.
Right.
But if some of them die, then maybe we still have some information for the record that we show that they die
Yep.
for some reason.
Yeah. So that's a really good thought.
It's a really short question. Any more?
Yeah, so we just had another question about, could you explain to us a little bit more how haplotypes and the inventory-based system will work together? So like we said, basically what we really need from a haplotype research standpoint is we just need a bigger volume of data. And we need full data, right? We need to know what happens on every animal, because what we're running into right now is we know we're missing these homozygotes, right? But we're left trying to answer the question as to what happened to them, right?
Exactly.
Are they just not getting genotypes because you culled those calves early on, right? And you weren't going to take them any further on in life, so you didn't invest that $37? Or are they truly those embryonic lethals that we've seen the dairy industry put out, for instance, and what we're trying to focus on here in-house?
Yeah.
So if we don't have any more questions for tonight, we don't need to keep you guys any further, but we want to thank you guys for joining us tonight for our Focusing on Fertility webinar. It sure has been a great time spent with you tonight, and I appreciate you guys all tuning in. Just a couple of things to note. We hope that you can join us on December 1st at 7 p.m. Central Time for our Investing in Inventory Angus University webinar. Here we're going to have two of our esteemed colleagues, Jerry Cassidy, our Director of Member Services, and Chris Stoller, our Chief Operations Officer, basically come down here and stand right here and actually explain a little bit more about that inventory-based reporting system and just the steps that you need to take as breeders to get involved in that. So with that, we'll sign off. Once again, thanks for joining us this evening and have a good night.
Thank you.